All right, so get ready for a deep dive into genetics today.
All right.
We're going to be talking all about how a rare condition in Lancashire Heelers can actually help us learn more about a human disease.
Wow.
And the article we're looking at is from PLOS Genetics. It's called IP3 receptor depletion in a spontaneous canine model of Charcot-Marie-Tooth disease 1J with amelogenesis imperfecta.
Okay.
Quite a mouthful.
Yeah.
But we're going to try to break this down to a doctor's level of understanding today.
Sounds good.
So let's get right to it.
All right.
Picture this.
Okay.
We've got a group of Lancashire Heelers,
Okay.
happy and active.
Uh-huh.
But they have one interesting thing about them.
What's that?
Their adult teeth have enamel defects.
Oh, interesting.
Yeah, so we're talking discoloration, thin enamel, some cases even exposed dentin.
Oh, that sounds like it would be painful.
You would think so, right?
Yeah.
But surprisingly, the dogs didn't seem bothered by it. Their activity levels were normal, and any nerve issues that they did have were subclinical, meaning they were only discovered through specific testing.
Okay.
Now, here's where the genetics get really interesting.
Okay.
Researchers used pedigree analysis, homozygosity mapping, and next-generation sequencing to figure out what was going on.
Wow.
And they pinpointed the problem as a homozygous nonsense variant in the ITPR3 gene on chromosome 12.
Wow. Okay.
Yeah, so homozygous just means both copies of the gene, one from each parent, have the same variant,
Got it.
and then a nonsense variant.
Okay.
It's kind of like a premature stop sign in the gene's code,
Oh.
which results in a shortened and often non-functional protein.
Okay.
And that's where the trouble starts.
Okay.
The ITPR3 gene basically provides instructions for building a very important protein called the IP3R3 protein,
Right.
which is one of 3 receptors responsible for regulating calcium signaling within cells.
Calcium signaling. That sounds important.
It is. It's super important. Calcium is like the signaling currency of our cells. It's vital for pretty much everything. Muscle contraction, nerve function, hormone secretion, you name it.
Hey.
Disrupting it can have all sorts of effects.
So this variant in ITPR3 messes up that IP3R3 protein.
That's exactly right.
Okay.
And figure 3A in the article actually shows us how the canine variant results in a truncated protein,
Right.
missing essential parts for proper function. The transmembrane domain and the C-terminal channel pore region are gone,
Oh, wow.
which severely impacts its role in calcium signaling.
I see.
And this brings us to the human connection.
Okay.
These dogs are a natural model for Charcot-Marie-Tooth disease type 1J, or CMT1J for short.
Okay.
It's a rare inherited neuropathy,
All right.
and it causes progressive muscle weakness and reduced nerve conduction velocity,
Okay.
just like what was observed in the dogs.
Oh, interesting.
And Table 1 actually highlights the range of nerve conduction velocities in both the dogs and human CMT1J patients.
Yeah.
And it shows striking similarities.
So by studying these dogs, we can gain insight into this human disease.
Exactly. It's a really powerful tool for understanding the disease and potentially developing treatments for both species.
Oh, wow. That's fascinating.
But the connection doesn't stop there.
Really?
These dogs can also help us understand why those enamel defects occur.
How so?
Well, you see, calcium is also essential for enamel formation.
Oh, okay.
It's crucial for the growth of hydroxyapatite crystals,
Right.
which give enamel its strength.
Okay.
So disrupting the calcium signaling,
Right.
as we see with this faulty IP3R3, it directly impacts that process.
Got it.
And it leads to amelogenesis imperfecta,
Okay.
which is a group of disorders that affects enamel formation.
So this one gene, ITPR3, has a hand in both tooth development and nerve function.
You got it.
That's incredible.
It is, and there's another layer to this whole thing.
Oh.
The study found that the affected dogs had reduced levels of not just IP3R3, as expected,
Yeah.
but also IP3R1 and IP3R2,
Oh.
the other 2 IP3 receptor subtypes.
So messing with one receptor affects the others.
Yeah, it's kind of crazy.
Yeah, how is that even possible?
That's the burning question, right?
Right.
It suggests this level of co-regulation between these subtypes that we didn't fully appreciate before.
Okay.
And one hypothesis is that the truncated IP3R3
Right.
being dysfunctional
Uh-huh.
might trigger this cellular quality control mechanism
Okay.
that degrades all 3 receptor types.
So the cell tries to clean up the mess, but ends up getting rid of the good receptors along with the bad.
You got it.
Wow.
And this finding has significant implications because it suggests that targeting one IP3 receptor subtype could have broader effects on calcium signaling than we previously thought.
Oh, wow.
And they found evidence to back this up.
Okay.
They looked at the dog's fibroblasts, cells found in connective tissue,
Okay.
and figures 3D and 3E in the article actually clearly showed decreased protein levels of all 3 IP3 receptors in the affected dogs.
Okay, so they've shown the protein levels are down. But how did they confirm that calcium signaling was actually affected in these cells?
They conducted functional calcium measurements using a few different techniques
Okay.
to measure how the cells responded to stimuli that triggered calcium release.
And what did those measurements reveal?
Well, one method they used was this ratiometric calcium indicator called FURA2AM,
Okay.
along with a fluorescence microscope.
All right.
And this allowed them to actually track the changes in intracellular calcium levels in real time.
So they could actually visualize how the cells were responding.
Exactly.
Cool.
They exposed the cells to ATP,
Okay.
a molecule that triggers the production of IP3,
Right.
which then activates those IP3 receptors,
Okay.
releasing calcium from intracellular stores.
So if those IP3 receptors aren't working properly,
Yeah,
you'd expect to see a weaker calcium signal.
precisely. And that's exactly what they found.
Interesting.
Figure 4A shows a significantly reduced response to that ATP stimulation in the affected dog fibroblasts compared to control cells.
The calcium signal was weaker in terms of peak amplitude and the overall area under the curve.
But how do they know this wasn't just because the cells had less calcium stored to begin with?
That is a very good question.
Yeah.
To rule that out, they measured total releasable calcium using a calcium ionophore called ionomycin.
Okay.
And this essentially bypasses those IP3 receptors
Okay.
and just releases all the calcium from those intracellular stores.
And what were the results of that?
No difference between the affected and control cells,
Interesting.
as you can see in figure 4B.
Okay.
This confirmed that that weaker response to ATP was really due to that impaired IP3 receptor function,
Okay.
not a lack of stored calcium.
Okay, so those FURA2AM measurements
Yes.
pretty clearly show that those IP3 receptors weren't working as they should.
Right.
But did they use any other techniques to confirm this?
Yes, they did.
What else did they do?
They used another method.
Okay.
A drug screening system called FDS-SUSEL
Okay.
allows for high-throughput analysis of calcium signaling.
And did they use ATP as the stimulus again?
They did, and the results were consistent
Okay.
with those FURA2AM measurements.
Got it.
The affected dog fibroblasts showed that significantly reduced response to ATP stimulation.
Okay.
But they took it a step further with that FDS-SUSEL.
Oh.
They also measured calcium release using thapsigargin,
Okay.
which is an inhibitor of the Charcot pump.
Can you explain what that Charcot pump does?
Certainly. So it's responsible for pumping calcium back into the endoplasmic reticulum,
Okay.
or ER, where a lot of the cell's calcium is actually stored.
Okay.
By blocking this pump, they could assess how much calcium was specifically in the ER where those IP3 receptors are located.
Oh, okay.
And what they found was that those affected dog fibroblasts had a significantly lower amount of calcium stored in the ER compared to the control cells.
That's interesting. Earlier you mentioned that there was no difference in total releasable calcium
Right.
between the two groups.
Yeah.
But now we're seeing less calcium in the ER of the affected cells.
Yeah.
How do we reconcile those findings?
That's the crux of the matter, isn't it?
Yeah.
It really suggests that there's more to this than just impaired IP3 receptor function.
Okay.
Disrupting IP3R3 might also be affecting the ER's ability to store calcium properly in the first place. While this specific paper doesn't dive super deep into those mechanisms,
Okay.
it certainly opens up a really intriguing avenue for future research.
It's like we've uncovered this hidden piece of the puzzle.
Exactly, but even without fully understanding that ER storage issue,
Yeah.
this research has already provided some really valuable insights.
Like what?
First and foremost, it reinforces that ITPR3 is a key player
Okay.
in both enamel formation and peripheral nerve function,
Uh-huh.
not just in humans, but in dogs as well.
So studying these dogs can teach us about human and canine health.
Absolutely.
Cool.
And these Lancashire Heelers
Yeah.
are a spontaneous model,
Yeah.
meaning they naturally develop this condition,
Right.
which makes them even more valuable for studying CMT1J and potentially other related conditions.
What makes a spontaneous model so special?
Well, a lot of animal models are created by introducing specific genetic mutations.
Okay.
While they're useful, they may not fully capture the complexity of a disease as it occurs naturally.
Right.
Spontaneous models like these dogs,
Yeah.
they provide a more holistic view of that entire disease process.
It's like studying the disease in its natural environment.
Exactly. And this gives us more relevant and potentially translatable findings.
So this research goes beyond just CMT1J then?
Yes, it really has broader implications for our understanding of calcium signaling in health and disease.
Okay.
So we've seen that calcium's involved in this wide range of cellular processes.
Right.
And those IP3 receptors, they act like these gatekeepers, right? Controlling calcium release from the ER.
Okay.
Any disruption to their function can have these widespread consequences.
And this study is a prime example of that.
Precisely, we're seeing impacts on both enamel formation and nerve function. Two seemingly unrelated systems.
Yeah.
It really underscores how interconnected those cellular processes are.
Right.
And the importance of tight control over calcium signaling.
It's a delicate balance.
Indeed it is. And this research gives us a glimpse into what happens when that balance is disrupted. The co-regulation of IP3 receptors.
Right.
It adds another layer of complexity to it all.
Yeah.
Targeting one subtype could have broader effects than we previously thought,
Okay.
impacting the function of others.
Right.
This could be crucial for developing targeted therapies.
So there's still a lot to learn.
Absolutely. There are many unanswered questions and exciting avenues to explore.
Where do we go from here?
Yeah.
What are some of the key areas for future research?
Well, we've uncovered a lot about these IP3 receptors and their role in calcium signaling. But where do we go from here? What are some of those next steps in this research? What are the questions that still need to be answered?
Yeah, what are like the main mysteries that we still need to solve?
Well, one of the big ones is that potential ER storage issue. We need to figure out exactly what mechanism is at play there. Is this a direct result of that truncated IP3R3 protein, or is there something else going on? Is there something else contributing to that ER's reduced ability to store calcium?
So we need to understand how that protein, like the messed up protein, is affecting things.
Exactly. Like, is it interfering with other proteins that are involved in calcium uptake or release within the ER? Or could it even be changing the structure or the function of the ER itself?
Yeah, those are really important questions to answer.
They are, because answering those questions could really change how we think about IP3 receptors in cellular health and disease. And beyond the ER storage issue, we also need to explore the extent of this IP3 receptor co-regulation.
Right, like how common is this?
Yeah, is this a universal thing, or does it just depend on specific cell types or conditions?
Yeah, great question.
It is. And how exactly is this co-regulation happening? Are there specific signaling pathways or molecular interactions that mediate it?
Right, that's fascinating.
Yeah, and these are all areas that are really ripe for investigation.
Definitely. And I feel like this opens up a whole new world for targeted therapy development.
Oh, absolutely. Imagine if we could fine-tune calcium signaling by just selectively modulating the expression or activity of different IP3 receptor subtypes.
Yes.
That could revolutionize treatment for a whole range of conditions.
Wow.
And of course, we can't forget about these Lancashire Heelers and just how incredibly valuable they are as a model for human disease.
Yeah, they've been so insightful.
They have. And by continuing to study these dogs, we can pave the way for new treatments, not just for CMT1J, but for other conditions that are linked to calcium signaling dysfunction. It's a real testament to the importance of studying animal models and just how much we can learn from our animal companions.
Absolutely.
This whole deep dive has been so fascinating. We've seen how just this one gene, ITPR3, can have such far-reaching effects, influencing both enamel formation and nerve function. And we've uncovered this really complex interplay between these IP3 receptors, calcium signaling, and potentially even ER function.
It's wild how it all ties together.
And it all started with these Lancashire Heelers and their kind of unusual dental issues.
Right.
It really just highlights how scientific discoveries can come from the most unexpected places and just how interconnected human and animal health truly are.
It's true. You never know where the next big discovery is going to come from.
Exactly. And it's a reminder that there's still so much more to learn and discover in this field.
I'm excited to see what the future holds.
Me too. The future is bright, and I can't wait to see what unfolds. From Vet NeuroJar, keep those minds inspired, hearts light, and tails wagging.